Hi, thanks for watching. My name is Guadagnin, and I'm a PhD student at the University of Illinois Urbana-Champaign. And today I'm going to talk about how feeding rumen-protected lysine during the postpartum can have an impact on uterine immunity. To better present the poster to you today, I'm going to split the poster in sections. I'm going to start with the introduction, objectives, and material methods, and then we're going to move to the results, conclusions, and acknowledgements. So first, as an introduction, we know that after calving, almost all of the cows are at risk of developing reproductive tract inflammatory diseases such as endometritis or metritis or even just purulent vaginal discharge. And immune events need to occur in a synchronous process with the metabolic changes that those cows undergo from gestation to lactation in order to prevent these reproductive diseases to happen. So throughout the transition period, those cows are in what we call a negative energy and protein balance, because this is a result of those cows not consuming the right amount of nutrients that they require to meet their demands. And this is usually associated with the development of metabolic disorders, but also uterine diseases. Because when the cow doesn't have the amount of energy and protein that they need, there's going to be also effect in an intracellular level. So for example, when there is a decrease in the intracellular energy source, this can lead to alterations in an inflammatory response and increase the risk of reproductive tract diseases. Also, in this first graph here, we can see a comparison of uterine inflammation according to the days relative to calving. So we can see that healthy cows usually have a very robust immune response in the early days after calving to fight the uterus contamination that happens in the early calving. But then that immune response is robust and effective. Therefore, the inflammatory response is resolved in a certain amount of time. And when it progresses, when we have some kind of disruption in that inflammatory process, then it becomes a problem for those cows. So therefore, we aim to determine the effect of feeding rumen-protected lysine pre and or postpartum on mRNA gene expression profiles of endometrial samples of hosting cows. We also wanted to evaluate those samples for morphology. In order for us to do that, those 75 cows were enrolled in a trial that started at negative 28 days, which are 28 days prior to the expected calving dates. They were assigned to one of 4 treatments, which consisted of either feeding lysine pre and postpartum, feeding lysine pre-partum but not post, feeding lysine just postpartum but not pre, or no feeding lysine at all. So therefore we have L for lysine and C for control. So now moving to the results. So in the first figure of this slide, we have the results for morphology, and we can see in the graph A that the number of cells per uterine gland was affected due to the treatment, right? We had a tendency for cows that received lysine postpartum to have higher number of cells per uterine gland. On the graph B, we can also see a tendency for effect on the glandular epithelial cell height, where cows receiving control diet postpartum tended to have a greater height. So at the bottom of the slide, we have the figure 3, where we can see the mRNA gene expression for those samples as well. And we can see that feeding lysine pre and postpartum increased the mRNA gene expression of interleukin 10, as we can see in the graph A. And feeding lysine postpartum tended to increase the mRNA gene expression of mucin 1 and mucin 4, which are mRNAs that encode glycoproteins that are involved in the epithelial protection and renewal. So to wrap up, in conclusion, feeding rumen-protected lysine postpartum impacted uterine morphology and immune responses, as it tended to increase the number of cells in the uterine glands and also increased the expression of mRNAs that are encoding glycoproteins involved in the protection and renewal of epithelial cells, and also increasing an important anti-inflammatory cytokine, which is interleukin 10, mRNA expression. So with that, the authors would like to extend a special thanks to the Dairy Focus team, the undergraduate students, and the staff of the University of Illinois Dairy Research Unit for helping through this study. And also we would like to thank Ajinomoto for funding this project.